So I appreciate the opportunity to present our work. It's a collective piece of work that I'm going to talk about that represents many groups on the campus and then individuals throughout the country and actually in the world. What I'm going to do is talk about bacterial quorum sensing in the minimal context, and then in the broader context talk about that in a way in which we're trying to develop tools that would allow us to manipulate the bacterial microbiome and understand how bacteria talk to each other and change their phenotype as response to environmental cues. And it's important to put this into an engineering context because there are very few tools available that allow for discrimination of biological activity at the length scales and time scales that those cells function at. And so let's just march right into it. So there's a medical motivation for biological and bacterial quorum sensing, which is elucidated here on the screen. So these are some pathogens that most everyone's heard about. And it turns out that if you're not familiar with quorum sensing, the bacteria talk to each other, if you will. So they secrete small molecules that then are perceived by those cells as they grow in cell density. And so Staph aureus secretes this peptide-like molecule, and then as the Staph aureus appreciates in cell number, then the gene transcription pattern takes on a collective behavior that is unlike the individual behavior prior to a high cell density. EHEC the same way. This is an autoinducer, AI2, which is the signal molecule for many gram-negatives. And there's another family of autoinducers, AI1, that again is prevalent in gram-negatives. And these mediate intracellular signaling, so bacteria that talk to each other and exclusively to themselves. In this case, it's highly specific. In this case, it's not so. And so the concept is that, and what emerged about a decade and a half ago now, was that these communication networks that are mediated by these small molecules mediate virulence in these pathogens. And so prior to that, the world of quorum sensing was really all about the marine organisms, Vibrio fischeri and Vibrio harvii, where it sort of sat for about 15 years or so. But then what emerged on the medical scene was that the attachment genes of certain pathogens, EHEC for example, were regulated by this communication network. And so normally we take an antibiotic to kill the bacterium that's a pathogen. The idea here would be that instead of taking an antibiotic that would kill the bacterium, you would take a drug or take a molecule or somehow mediate the virulence by affecting the communication network rather than killing cells. So normally when you take an antibiotic, the cells die, and that leads to a selective pressure for the emergence of resistant strains, hence all the multiple drug-resistant strains that are out there. And so the idea here is knock out the communication between the bacteria, and then actually we might avoid the disease that's affected. And so one of the things that we've been trying to do is develop a series of tools that allow for the communication that is between biological systems and communication between electronic device systems and microfluidics, and bridge this kind of a gap. And so we've been very involved in understanding the molecular genetics of quorum sensing in E. coli. We got into this, now it's been almost 15, 20 years ago, because we made recombinant proteins in E. coli, and I had one graduate student who's now up for full professor at Cornell who wanted to look at E. coli fermentations and said, okay, maybe this communication system is happening in E. coli fermentations at high cell density. We didn't know that, and so he asked the question, indeed it was. And then the challenge was, though, that as an engineer, no one cared about lab rat E. coli. It was all in the medical community. And so we wanted to understand what happened in E. coli K12. And so we spent a lot of time, it's now been in the early 2000s, elucidating how this signal mechanism works. So I know you can't see this, and it's not really that important. So basically, the autoinducer 2 is secreted by a 2-enzyme pathway that I'll show in the next slide. And it's brought in through a transporter. The signal molecule is phosphorylated by a kinase. It binds to a transcriptional regulator, which is then comes off the operator promoter site here, and that then de-represses the synthesis of these different genes. And the genes on the right, which you can't see, are actually transporter genes. So the accumulation of the signal molecule in the extracellular milieu, it goes up, and then as it comes in, it then comes in at a higher rate as these genes are de-repressed. So here's the pathway for this particular signal molecule. It has these enzymes PFS and LuxS that go from S-adenosyl homocysteine and prior to that S-adenosyl methionine. So for those of you that care, this is the activated methyl cycle. So where metabolism puts one carbon onto any carbon backbone, this is the carrier. So this is central in carbon metabolism. And then the signal molecule comes out as a response to this carbon metabolism. In eukaryotes, you and I, we don't have PFS and LuxS that make the signal molecule. We have an SAH hydrolase that generates homocysteine and adenosine. And so that makes these enzymes key targets for quorum sensing antimicrobial activity, because they're in bacteria and they're not in humans. And so if you want to knock out quorum sensing activity, you might target these enzymes. And so what I'm not going to really talk about today is the work that we've elucidated in the last couple years. That's with Sergei Sukharev and Herman Sintem's group in chemistry, where we generated a series of C1 alkyl analogs to this signal molecule, the dipentane dione. And just to give you the upshot of some of that work, it turns out that these analogs go through the same signal transduction cascade, get phosphorylated, and then bind to the transcriptional regulator, but it's a competitive inhibition. And so by occupying the regulator, you can quench the activity, and this is commonly referred to as quorum quenching. And so this might be a surrogate for the communication pathway, and actually could interrupt the communication pathway. So actually, fortunately for Herman and the group, these are now patented, or soon to be patented, I believe. So what I really want to spend most of the time on is a technical approach to sort of understand the communication network in a way that allows us to understand the physiology of the system being many different types of bacteria that talk to each other. This is something that's not easily accomplished. So we've termed a word biofabrication, that is the convergence of biotechnology and microfabrication. If you think about the paradigms of microfabrication, which is on the left-hand side here, microfabricated devices, electronic devices, communicate via electrons and photons. So we have absolute reproducible. When you hit a 9 on your cell phone, you get a 9 that shows up in the little window. When you hit a protein with an AFM, it may make some functional change. It may not, depending on what the context is. There's no context dependence here. Communication back in biology is mediated by a different means, ions and small molecules. And so the question is, how do we actually bridge this gap? And so a large part of our effort is with Greg Payne's group, Gary Rublov's group, and Reza Ghazi's group on trying to develop strategies that allow us to reconstruct biological functions in microfabricated devices. And a large part of that is mediated by an interface. So we want to develop an interface that actually can be biofunctionalized, if you will, assemble biological components into a device that might have microcircuitry. And so we use polysaccharides as an interface. And this describes a polysaccharide called chitosan, which is a polyglucosamine. It has an amine group that has a pK of around 6.5. So if you actually submerge an electrode, which you don't like to do with your cell phone, but we can do that here, and you generate a pH gradient by having a negative bias on the electrode. So this little yellow thing is an electrode under a microfluidic channel. That causes a raise in the pH, and chitosan that's in the vicinity, the amine group is going to be deprotonated, so it has no charge. When it has no charge, then those chitosan molecules can assemble with each other and form a gel or a film, depending on how that charge is applied to that aqueous solution. If it's protonated, it's soluble. Okay, so we're really looking at controlling the transition between soluble and insoluble. And once it's put down onto this electrode with the spatial resolution that's defined by the microelectronics. So feature sizes in microelectronics are continually getting smaller and smaller. And so the concept here is let's develop ideas that will allow us to piggyback onto that and put biological components into the device. So we've done this with a series of different kinds of polysaccharides and biopolymers. The second one is alginate. Calcium alginate has been known for a long time for immobilizing enzymes, immobilizing cells, etc. And it works in the opposite fashion. Here, alginate it complexes with a calcium ion. And the calcium ion, when it's complex, forms this egg box-like structure. And so here we have an anodic signal to an electrode, and instead of having just a pH change, here we have inorganic calcium carbonate that's normally insoluble, and then it gives an acid, and that solubilizes the calcium carbonate to get the calcium ion, and it complexes and forms alginate on the electrode. And so there's a series of other works with gelatin and agarose that I'm not going to talk about, but the idea here is to build a system that allows us to assemble biological components into a device. Well, when you can make use the pH, you can make a pH gradient using electronics. You can also make a pH gradient using microfluidics. The same technology should apply. And so here, Xiaolong Luo in our group has created a microfluidic channel, if you will. There's two fluid channels that are convergent, and while it's a green film here, pretend it's not there. So it's just an intersection of two fluids that can hit each other, but they don't mix because it's a low length, small length scale. Those are laminar flow streamlines. And so in one case we have a buffer solution at pH 10, and then in the other case we have chitosan at pH 5. And so at the interface, pH 10 is going to lead to the insoluble formation of a film that actually transverses from this point to that point, from the top to the bottom of this microfluidic channel. What is that good for? Well, that's good for building networks of cells, proteins, etc. On this interface, and then it allows for the superposition of different types of cells or fluids on either side of this membrane. Okay, so hold that one thought for a little bit. So here, once we had this chitosan film, we built then an assembly of alginate and cells. So here, calcium chloride, the calcium ion's free, is introduced, and alginate and cells are introduced on this side. So the calcium then will diffuse from this channel to this channel, complex the alginate, and capture the cells. And so in this way, you can build networks or layers or biofilm mimics of bacterial cells or whatever cell that you wanted to apply. And so here's the red line is the chitosan film. The green pattern here is a series of bacterial cells that make green fluorescent protein. You can switch from calcium on the top to alginate, and alginate on the bottom, to calcium on the bottom and alginate on the top. The calcium goes up, and then you can layer another set of cells here. And then Xiaolong used all sorts of different techniques and gimmicks to try and make layered cells that have different characteristics. In this case, you have blue cells that make a blue fluorescent protein, green fluorescent protein, and a red fluorescent protein. And what's neat about this is that you can layer cells very rapidly in minutes. They're functional, they're active, they have media, and it's absolutely a system that allows you to control the flow of media and components to the cells to keep them viable for a long period of time. If you take citric acid and run that through these channels, you can peel the layers off and then look at those cells under in situ techniques that you may have offline. And so here we just demonstrate the concept of controlling quorum sensing that's always at high cell density. So the common thought of bacterial quorum sensing is that it emerges as cell density increases. And really what it is the signal molecule that actually increases. And so here, what Zhelong did is set up two populations of cells on the top and bottom of a chitosan layer. The top layer of cells make green fluorescent protein when they're making autoinducer 2. And the bottom set of cells makes red fluorescent protein when they're perceiving the autoinducer 2. And so what's shown here is a series of slides where the flow rate of media in the bottom channel, LB media, is fixed for a long period of time. And so this is after about 18 hours at different flow rates, and the flow rates are increasing. So at low flow rate, all the cells are responding because AI2 is diffusing from these cells to these cells, and they're making red fluorescent protein. At the highest flow rate, these are making autoinducer 2, but what's happening is the media, which has no autoinducer 2, it's just fresh media, runs through this gel, this hydrogel of cells, and this effectively dilutes out that autoinducer 2, so those cells no longer respond. And so the concept here is that in a system, you can control the bacterial quorum sensing behavior, which is a collective behavior, by just controlling the flow rate of a media that has or does not have that autoinducer. Why is this important? Well, ultimately, what we'd like to do is understand the digestive tract. The digestive tract has flow that's pulsatile, that's complex, and you've got to be able to think about ways in which bacteria respond to those environments as well as how it interacts with the epithelium in the digestive tract. So this method then, if you have optical transparency, you can elucidate the phenotype of cells as it responds as they respond to the autoinducer as it travels through this gel. And so this set of images here is taken by a series of cuts from top to bottom of the red layer. And so the autoinducer 2 is highest at this interface, and then it's lowest where the media is going through. And so here is the interface, and here's where the media goes through. And you can see by the different profiles that and these are resolved colonies of cells, they're not individual cells, but you can see here how the cells respond to the gradient in the small molecule in a biofilm that was set up artificially. I don't know of any other techniques that allow one to rapidly identify cell behavior at this scale and at this time, and that's why I'm really excited about this, because we can start to look at how the cells respond. So another question that we have then is how can we manipulate the quorum sensing with sort of nanoscale resolution? And so I turn to another way in which we might modify the behavior, and this is by localizing the synthesis or degradation of the autoinducer on a cell. And so this concept was published in Nature Nanotechnology, a biological nanofactory, if you will, in 2007. The concept was, it's for drug delivery, the drug delivery world, is have a factory, if you will, that has a synthesis module where instead of taking a drug and loading it up on a nanoparticle that's targeted to a cancer cell or a cell, where the therapeutic threshold of that drug has to be on that particle or those particles, make the drug at the site by resources and raw materials that are localized. That's the concept. So you have a targeting module that sends it to a target cell, a sensing module that knows when there's the precursors there, it makes the output, and then, like Mission Impossible, self-destructs miraculously and it goes away. That's the concept. Rohan Fernandez in our lab decided, well, that's interesting, can we try to do that? And so what he did is he took those two enzymes, if you remember, that made the autoinducer 2 in E. coli, PFS and LuxS, and added a protein G domain. This is now a 3-domain fusion protein expressed and purified in E. coli. The protein G binds to the Fc region of any antibody of interest. And so that antibody would be an antibody that targets whatever you want it to target. And so by doing that, you localize the nanofactory, if you will, on the target cell. And so what we did in this paper was to target E. coli in a mixture of E. coli and Salmonella. And you got a quorum sensing response in E. coli and not in Salmonella, and vice versa. So the concept here is then, can we manipulate the quorum sensing response on an individual cell basis? And that's at least one technique that incorporates this kind of concept of fabrication. But you're not going to be able to take that fusion protein and ingest that and have it do anything in your GI tract. So then we turn to a protective layer. And this is work with Srini Raghavan, who's in chemical and biomolecular engineering, and Greg Payne, where we took these nanofactories, if you will, and immobilized those into this alginate chitosan capsule. So here it's a different construct than on the microfluidic system, but it's similar. So the green is the alginate. So we have calcium alginate, basically drop alginate into a solution of calcium, and it makes these little drops. Those are not really all that stable, and they will not hold a protein that's embedded within that. So things can leak in and leak out. We then immersed this into a chitosan capsule that was then cross-linked, and the chitosan is the red. And there's a confocal image of the red. I don't know if you can see that, but this is a cutout. It's a very uniform, nice film around the outside of the alginate. And what that does is it confers a semi-permeability to this system that allows for the nanofactory to stay inside and small molecules to diffuse in and diffuse out. And so the concept here is to make these capsules, if you will, and immerse those into bacteria, provide the reactant SAH, and make autoinducer 2, and then have that modulate the bacteria that are localized. So here's a couple of slides that show some of the data for this system. The first one, you ask the question whether or not the nanofactory secretes out of the capsule and makes autoinducer 2 if you add this substrate. And the answer is no until you puncture the nanofactory, or puncture the capsule, and then all the contents spill out. And then the other question is whether or not if you just add SAH to the intact capsules, can you make autoinducer 2? And of course the answer is yes. And so then you ask the question whether or not if you take these capsules and put those into bacteria, can you modulate the signaling behavior of the bacteria by providing the substrate? And so in the left panel, what we have, these cells actually are LuxS minus, so they do not make autoinducer 2. And so by immersing these capsules in and adding SAH, we're making autoinducer 2, and they should respond. And in fact, they do. That's not surprising. What is interesting, though, is that wild-type bacteria, when immersed with these capsules, can respond also. So already these cells in the black, W3110, communicate and are making green fluorescent protein in this case. But when we put the capsules in, we make more of this autoinducer 2. They actually respond by changing their gene expression patterns. So one of the things that I've hopefully conveyed so far is that we have these networks of communicating cells, and these cells, by quorum sensing, align themselves in their behavior. And that's typically the case of synthetic biology. So this is my concept of the biosynthetic capability of a cell. There it is. And the question is, how do we manipulate that in synthetic biology? So typically what will happen is we have a linearization of the system, which is a gross exaggeration, and we apply constraints, and then the optimal solution of those constraints is normally at the boundaries and the intersections and of the constraints. So in synthetic biology, the idea is to rewire genomes and have cells make something they normally wouldn't make. And so most of the work in synthetic biology is on small molecules. There's a lot of it on biofuels right now, where you want to make A and B, but you might also want to make C, and you put biocatalytic activities and enzymes and pathways into the cells. And in this kind of paradigm, all the cells might make a biodiesel, and they all operate identically, and they all are optimized identically. And so a different concept of synthetic biology is to view the cell as the product and ask the cell to do something that's different than it normally does, recognizing the individuality of the cell. So I have my animated graphic here. That's the cell, and that's our new product. So here's an analogy. Can we ask cells to do something completely unorthodox? Can we ask them to be like a dirigible and go somewhere, take an action, and then leave? So in order to do that, we have to recognize, what does the dirigible have? Motility, docking capability, control modules, and they take an action. Can we have cells do that same kind of function? And now I'm going to tie this somehow into the GI tract in about 5 minutes. So the idea here is, can we get bacterial cells to recognize cancer cells from non-cancer cells? And the distinguishing features of cancer cells are typically that they have some marker on the outside. And in this case, EGFR is a marker for certain tumors, and that would be something that we'd want to capture and recognize. So the idea would be, can we build cells that would recognize the EGFR, and then based on the concentration density of EGFR on the cancer cell, which is not high on the normal cell, take an action by making a drug. So here the nanofactory really is the cell, not a little actual device. And so to do this, we set up a system that's like a test track, using the surface assembly techniques of chitosan deposition, electrodeposition, and alginate electrodeposition. We added avidin to plates and coated avidin, and then added nanofactories that have anti-avidin. So this has the two enzymes that will make AI2, but it targets to avidin. And so if you have avidin coating plates at increasing concentration, you should expect nanofactories to bind in some sort of Langmuir isotherm. Then if you add the substrate, you should expect autoinducer 2 to follow the trace of the nanofactories. Then if you add cells in the surface above these nanofactories, they should respond to the autoinducer 2, and those appear to respond in the same sort of proportionality. So the idea here is, can we build a test track that will allow us to put the cells down, recognize cells, and have those cells target the EGFR? So the next question was whether or not bacterial cells could respond when they're surface targeted. They need to respond when they're bound to the surface. So we did the same thing here. In this case, we made cells that have protein G on the outer surface, and then load those cells up with an anti-EGF or anti-avidin antibody, and stick cells onto those, and then add autoinducer 2 that's in a high concentration to those cells, and ask the question whether or not we can have a linear increase in the amount of cells, like we had, at least at low concentrations of avidin, a linear increase in the capability of making the autoinducer 2. So both of these seem to work. Then we asked the question whether or not, if cells are bound to these, if bacteria are bound to the cancer cells, can they recognize a difference in the receptor density? And so here we loaded up different concentrations of these 15 B cells, these are the cancer cells, and then docked the bacteria in the green line that are loaded with the anti-EGFR, so you can dock bacteria onto the cancer cells. Previous slide was on a surface. And then we asked the cells to make autoinducer 2 by loading nanofactories onto the cells. So it's a complex assembly here. At low cell density and low nanofactory concentration, we didn't have a lot of autoinducer 2, and we got a jump in the amount of autoinduced or of red fluorescent protein at higher concentrations of cells. So indeed, we had the no-go decision responding as we would like to see. So when you think about the system, we have a recognition capability of putting cells down onto a cancer cell. We know that if they have different amounts of autoinducer 2 when they're exposed and bound to the cancer cell, they will go through this quorum sensing switch. We don't know how to get the cells to the cancer cells, though. It turns out that autoinducer 2 is a chemoattractant. And so here we have a plot of cells that actually migrate to autoinducer 2. So all this is a common transwell apparatus, if you will, on the upper left. It's like a double boiler, if you will. So you have a vessel with an insert that has a semi-permeable membrane, and you have autoinducer 2 in the top, and bacteria in the bottom, and ask the question whether or not the bacteria will swim up and go into the autoinducer 2. And that just shows that they in fact do. So the cells will migrate to autoinducer 2, and then we generate cells that shift their gene expression to different levels of autoinducer 2. And so then you can put the whole system together, and here we had the cancer cells on an insert. They're loaded with nanofactory on the EGFR. So the cancer cells with nanofactory will make autoinducer 2 when supplied with the substrate. That autoinducer 2 will diffuse down into this chamber. Bacteria that are below will swim up, and then based on the threshold concentration of that autoinducer 2, will turn on gene expression. And so this is a demonstration of that. We have, in this case, this is the case where we have the 15B cancer cells and PBS. Here's where we have S-adenosyl homocysteine. The bacteria go up. They make autoinducer, the nanofactories make the autoinducer 2. And here we can see by the DS-red response, they switch on their gene expression when they've reached that threshold. And so what I'd like to end with then is sort of an overall approach, hopefully I've conveyed, is that we're trying to develop tools that will allow us to interrogate complex bacterial consortia in complex environments. So here we have a microfluidic system, or a series of systems that we can put different proteins and cells in different configurations, and we've developed different tools like this nanofactory that can resolve the placement of these signal molecules on individual cells. And then we've also thought about ways in which we could put those into a complex environment that will protect those nanofactories if you wanted to make the autoinducer 2 in a GI tract. So with that, I'd like to recognize especially my collaborators here in College Park, and the people that are circled are the graduate students, former graduate students that participated directly in this work, as well as my current group. Thank you very much. One question?
One question.